So basically, all of the topics that we covered yesterday were how to get from your very raw data at the output of your sequencer to an analysis-ready BAM file. So yesterday, we covered topics like in the realignment and base quality recalibration, and all of these steps that get you a BAM. So what do you do now with the BAM? And this is sort of the topic that we're going to talk about today. Now that you have your BAM, now that you have your recalibrated reads, your correct base qualities, what do you do with them? And the main objective in most analysis that we do is actually get to detect genetic variations. So if I have a set of samples, what mutations and what genetic variations do these samples have in the context of my research of interest? So this is what we're going to be covering today. The two sort of main workhorses that the GATK use for detecting variation are, first of all, Unified Genotyper, which is our legacy tool, and the newest, fanciest state-of-the-art tool, which is a Haplotype color that Ryan is going to talk to you about. So I'm going to start talking about Unified Genotyper in a second. So just to give you an idea of what Unified Genotyper does, Unified Genotyper is the tool that takes as an input a BAM, your recalibrated, already processed BAM, and outputs a list of genetic variants in your samples. It can work single sample, it can work multi-sample, it works for SNPs, it works for indels, as we're going to see in a little bit. So that's sort of like the main tool to actually get your genetic variants. In a second. If people actually sort of went to the tutorial and the hands-on session yesterday, at the end, we could see actually that through IGV, we could see the BAM, we could visualize it, and we could see the reads that were in some places that had like very clear indications of where a SNP was or where an indel was. However, sometimes it's very easy to actually see where these genetic variants are, and you can sort of see them by eye. But there are circumstances in which it's very difficult to ascertain whether there is some variation there. And that's why we need a little bit more sophisticated methods to actually deal with this that I'm going to show you in a bit. Would it make sense if we hook up my computer directly?
I'm not sure how to switch the computer.
Oh, okay.
I have
Okay. Okay.
Sorry about that.
Yeah, no problem. So as I was saying, sometimes it's very hard to actually detect or to distinguish this genetic variation from background noise, which is what I'm going to show you. And that's why we need a little bit more sophisticated models to process the data. All right. All right.
Sorry about that.
So as I was saying, we are calling variants here. And as I was mentioning, this is sort of a typical picture of what IGV, an IGV screenshot looks like. And as I was saying yesterday, sometimes we could see very, very clear variation, SNPs that covered like a whole sample, and this is a very deep sample. So it's very clear what's going on, and you don't need like fancy math. I mean, you can just by eye say, oh, there's a SNP here, or there's a deletion there. But real next generation sequencing data a lot of time looks like this, and it's very hard to figure out, well, what's real and what isn't. So for example, here in this location, you have this green mismatch in maybe something like 20 or 30% of your reads. So is that a real SNP? Who knows, right? I mean, it could be either way. So that's why we need a little bit more sophisticated analysis tools. And and the question that we're trying to answer is, well, is there a genetic variant in my data, or is this some random noise? So we have this large-scale inference problem. We use a Bayesian sort of framework to actually solve this. And as I was saying, we have two approaches. And I'm going to focus first on this initial approach, which is unified genotyper, which is very fast, but it has some limitations. So the idea of unified genotyper is that you sort of walk through the genome, and at each position in the genome that you're interested in, at each locus, you ask, is there evidence for variation or not? So that's sort of like the main approach. You walk and independently ask, is there evidence for that? Now, it turns out that this is very fast, but it has like some limitations, and that's why we came up in the past year with this new approach called the Haplotype Color, which does localize de novo assembly that Ryan is going to talk about later. Now, the unified genotyper, what it does, as I was saying, is that for each locus, so for each genomic position that you run it in, it's going to first determine the possible SNP and indel alleles. It's going to compute for each of the samples that you give it the likelihood of the data given all of the possible genotypes at a location. And then given these likelihoods, it's going to compute an allele frequency distribution to determine the most likely allele count. So, so this distribution tells you essentially what's the likelihood of having k alternate alleles for all of the possible k. And if there is enough evidence that there is actually a genetic variant there, then we will output a call. So I'm not going to go into like the deep details of the math, and if you're interested in the nitty-gritty of the analysis and the math, please come talk to us. We will be happy to explain this or to refer you to where you can find more detailed information. But essentially, the idea is that given your data, you form the likelihood of your data given all of the possible genotypes, and then we assume that all of the reads are independent, so we can decompose this just in a product of all the likelihood of each read given all of the possible genotypes. And then essentially, based on base rule, we compute the probability of each of the possible genotype. As I was saying, the reads are assumed to be independent, and this is sort of what the internal machinery does. Now, for SNPs, this is actually very easy because you just need to determine essentially for each of the locations that you're interested in, what's the probability of the data given that you have A, C, T, or G. And this is actually a very simple function of your base qualities. So for example, if you have a mismatch of the base qualities, then essentially the probability of having the data given a certain base is just the probability of error, which is given by the base quality or is related to the base quality. And then you can sort of decompose everything that way. Now, an important consideration is that for computing these likelihoods, we're sort of pre-filtering the data, and we're only considering the bases that satisfy some quality threshold. So, for example, we discard all of the bases that have a quality lower than the minimum quality. We discard reads that have like 0 mapping quality or a mapping quality lower than a certain threshold, etc. So we only consider sort of like the clean data that we have at a certain location. Now for indels are a little bit harder because first of all, you cannot list a priori all of the possible indels that you have. So the approach that we take in unified genotyper is that we only consider the alleles which might be present in the reads and that the aligner put in. So for example, by default at the BroadE, we use BWA. So if BWA actually opened a gap at a particular location, we will consider that for the indel discovery. So based on these indels that the mapper actually produced, we form haplotypes, and then we score the reads to these haplotypes with an HMM. And again, I don't want to go into like the deep mathematical details of how this is done, but if you're really interested on how we do this, please come talk to us. But this is actually a very standard thing that people do. So once you have these likelihoods for each of the samples, as I was saying, we sort of combine the likelihoods to see what is the overall evidence for variation at a particular site. So we take these likelihoods, we do this math that we call the exact model, which is just essentially based on the likelihoods, what's the probability of having a certain allele frequency across all of the allele frequency spectrum. We have a population frequency prior that we can apply. So essentially, at the end, we have this distribution of that tells us what's the probability of having a certain allele frequency, or to be more precise, an allele count of 0, 1, 2, etc., given your data. And based on this allele frequency spectrum, we determine whether we make a call or not. So in practical terms, how does this look? Well, Unified Genotyper is just another tool of the GATK that you give it a certain BAM or a list of BAMs or several BAMs at the same time, one or more samples. You have several parameters that you can tweak your output. So for example, you can choose to be more selective. So you can choose to have only as outputs like variants of really high quality that you're very confident in, or you can choose to be very sensitive and you can have more variants even if their quality is not that great. So we have a couple of knobs that you can turn, which are described in the documentation. And as an output, you have a BCF file. And this BCF is essentially a table that tells you all of the discovered variation and then for each of your samples that you have, plus some site-specific information. And later on in the hands-on exercise, actually, you will get the chance to actually run this and examine, based on some real data, what the output looks like. Now, as I was saying, this is sort of like our standard sort of older workhorse tool that we extensively use. But it has some limitations. The state-of-the-art tool that Ryan here developed is called Haplotype Color that he's going to describe right now, which is sort of like the tool of choice for more sophisticated analysis and which is more sophisticated than Unified Genotyper. So Ryan?
So I think now would be a time for questions about the Unified Genotyper, if anyone has them, for Guillermo.
Yes. Yes.
So when we code the genotypes, we need to know the prior of the population allele frequency. But if that's, for example, a novel, like the new variants that we are going to discover, how you assign the allele frequency?
Okay, so by default, we have a prior that you can modify. So the prior is based on what we think is like standard human genetic variation of like, 1 in 1,000 for SNPs or 1 in 10,000 for indels. But you can certainly change that. And that's another knob, another parameter that you can tweak. So you can modify that by a command line argument based on your data and based on what you think should be like different priors for populations, or if you're doing somatic analysis or whatever.
Or a different organism.
Yes. Mm-hmm. Yes.
What's the speed difference between the haplotype color and the unified?
So the difference between haplotype color and unified genotyper in terms of speed, it used to be 10 to 1. I don't know.
It's probably like 5 to 1 now. It's 5 times slower. And there's tons of parameters that you can do to play with trade-offs between runtime and sensitivity. I'll talk about that a little bit. Mm-hmm. The problem is you're doing a lot more work, so it's slower. Mm-hmm. It's also harder to parallelize, which makes it, depending on how your pipeline's set up, it's even slower than 5 times slower.
Sorry, so the haplotype is slower?
Yeah.
Yes.
We're already starting with the negatives. Let's talk about the positives.
Right.
Okay. Thanks. Yeah. Okay. So, like everyone's been saying, the haplotype color is a brand new fancy thing. I'm going to try to describe it by contrasting it with the unified genotyper. So, so the major difference between the unified genotyper and the haplotype caller is that the unified genotyper is sort of trapped in this per locus view. You come to a locus and you go to each one independently. That's why it's massively parallelizable, because you go to each locus independently. And you ask the question, what's the evidence for variation at this locus, using only the data that overlaps just that one locus. You make the decision for a locus 1, and then you move to locus 2, and you start from scratch and redo everything again. So the fundamental difference really is that we're no longer treating reads as these independent things, and we're looking in regions for variation instead of just independent loci, right? So when you come to these loci, you're asking yourself, what's the universe of possible alleles at this locus, right? That's the way the unified genotyper works. It enumerates all the possible alleles at this locus and then evaluates those alleles, evaluates the probability of those alleles in this Bayesian framework. And so what the haplotype caller is going to do instead is it's going to come to a region that it's a potentially variable region, in the same way that you saw reduced reads yesterday cut up these regions between variable regions and consensus regions. This is going to do the exact same thing. It's going to find the variable regions, and it's going to say, what are the possible alleles in these regions? And it does that through local de novo assembly. So it takes all of the reads in a region, puts them into a big bucket, and builds an assembly graph out of them. So, and then this graph is you're going to read off the paths in this graph, and those paths are going to be the potential haplotypes, and you're going to evaluate those haplotypes. So the way it comes up with possible alleles is by building this assembly graph, which is very different than just coming to the reads and trusting their alignment and asking what's the universe of possible alleles using just those reads. Does everyone see the contrast between that?
So is it a bit like what Indel realigner does, and that kind of shifts everything?
Sort of. The difference though is that the Indel realignment, it's using like the known indels, right? And that's its possible indels. Whereas this is building any number of possible indels based on the reads itself, right? So it's not trapped by using prior databases or the read data itself. It's using a bunch of reads in a region and building alignments out of that. That's the real difference, is that it's using a large region instead of a smallish region. What's up? That's exactly what it means. Yeah. Because you're not using the alignment of any read actually. So you're using its approximate placement. So we make this distinction between mapping and alignment. So the mapping of a read is like its approximate placement, where the alignment of the read is like its actual alignment with indels or not, and it's like precise location. So because of this, in the haplotype caller, you're not actually using the alignment of any read. You're simply using all the reads that are localized to a region and trusting its approximate mapping, which is pretty good. Aligners do a pretty good job of approximately mapping a read, but they don't do a good job of precisely aligning a read. So that's the fundamental difference. So you don't need to do indel realignment because you're not using the alignment of any read. Make sense? Okay. What's up?
That we still need to the realignment before the haplotype caller.
So if you, so no, you don't. It wouldn't matter, right? It wouldn't matter whether the reads are realigned or not.
So you mean if we want to call haplotype caller, we can just go from the multiplicate and then recalculation? Exactly.
Yeah. So you save a little bit of time by not having to do the indel realigner step, I suppose, but you lose it in the fact that the haplotype caller is going to be much slower than unified genotyper. What's up?
Is there any efficiency to be gained by moving this into the alignment software?
Potentially. Yeah, I think it's better to have like separate tools for separate jobs, right? You can't have the aligner do every possible thing. Because, for example, the aligners in general don't even use the quality scores. Like, they don't care about quality scores. And so I think it's probably better to have specialized tools for specialized analyses, What's up?
So I have a question. What's motivation for joining your method? Does this mean that unified genotyper didn't generate quality variants?
Yeah, see, that's the thing. It's troubling because we created the unified genotyper, and it works very, very well. But I'm going to try to show you that, in fact, it's not so good. So it does a lot of things very well, right? But the places, like things like more complicated indels, more larger indels, for example, it does a pretty poor job, right? Just because it's trapped in a per locus view, it has these limitations. And so the examples I'm going to show you in a few seconds are all examples where the unified genotyper does poorly. But in general, like for calling SNPs or for calling smaller indels, the unified genotyper does very, very well. And it's been our workhorse for a long time. So things always get better, right? What's up?
So is there a maximum size of this region, or do they actually settle?
So it's all like set by parameters to the algorithm. There's no like fundamental maximal size. I think by default, it works on regions of 300 base pairs.
So it can't be assembler, like assembler genome.
Oh, sure. It's not going to assemble an entire genome. No, no, no. It's local de novo assembly is the idea. And you can push that to thousands of bases if you wanted, but it's never going to do like a full whole genome assembly. It's just, we've sort of taken it's just a trade-off in terms of efficiency, right? So, any other questions? Lots of questions, yeah.
So maybe I'm jumping the gun, but do you know what's the accuracy level versus unified genotyper? What's the gain in accuracy?
Yeah, I don't know. I don't have any specific numbers. It's so hard to measure these things. I don't know. It's a lot better. I don't know what else to say. I don't have any like numbers. Sorry.
So just to complement that, for example, unified genotyper in 1,000 genomes, we achieved like a false discovery rate of like less than 1% for SNPs. So for SNPs, it's really, really good. For indels, our false discovery rate was maybe like 5%, 10%. But then, the other thing is that we didn't discover with unified genotyper any of these large variants that Haplotype color is now discovering. So in that sense, this is doing like infinitely better than that.
That's a much more analytical answer. Thank you. What's up?
So is there an easy way to stage these, tandem these things? You give the easy cases to unified genotyper, just mask those and give them.
Yeah, so that's not the way it works right now. Potentially, we should consider doing that sort of strategy in terms of efficiency, but we're still in the middle of developing the algorithm. I mean, it's very new. And so sort of efficiency sort of concerns come away later for us. So I think eventually we'll probably do a tandem thing like you're suggesting, where we call the unified genotyper first, and places, maybe it'll tell us when it's not sure, and then we'll go back and do the full shebang with the Haplotype caller. That's a good idea. It's not what we do right now, though. Right now, we just treat the two procedures as independent. We usually run both and just see what the differences are. Any other questions?
So, does error rate have a big effect on like the complexity of the burn graph?
I'm not sure what you're asking. Does what?
I guess I'm thinking in terms of like this assembly land. You try to error correct the reads so that you can get a cleaner graph. But if you're using the raw reads, which have the errors in them, is it complicated?
Yeah, I mean, so any error. Wow, I haven't even gotten to a single slide yet, but basically, I mean, any error in the read is going to show up as a path in the graph, right? And that's bad. So you create lots of paths in the graph. And so we have this procedure for pruning the graph. And that does an okay job, but it's not great. So you're right. As the regions get more and more complex, sort of, there's more and more paths. It takes longer. You have to evaluate tons of haplotypes. But what's going to end up happening, you'll see, is that we're using the same HMM that the unified genotyper uses to evaluate haplotypes, and that is what allows us to control our error rate, is using that HMM to evaluate the probability of a haplotype. And so it's simply a matter of computational trade-off, right? If you give it 1 billion haplotypes, it's okay. You just have to wait a long time, right? But the HMM is what allows you to evaluate the probability of those haplotypes very accurately.
So what's a minimum coverage then for trying to do this?
Well, it would just be, so the question is the minimum coverage. So we use it for 1,000 genomes 4x coverage, and we achieved very high accuracy. So I would say But I mean, the trade-off there is you have lots of samples, right? So it's 4x coverage, but you have lots of samples, and that's what allows you to build these graphs. So for the single sample case, we're sort of working with like down to even 15x coverage, and it looks fine. So I don't know. Is that an answer? Something like that. Anything else?
Actually, I've noticed that I was in a case where I artificially introduced errors into my BAM file.
Yeah.
So I had a very high error rate, and the indels didn't get called at all. Have you seen that before? With the traditional indel realigner and everything.
Is this with the unified genotyper or the Haplotype column?
Unified genotyper.
Well, so one way that the indels wouldn't be called in unified genotyper is if there isn't like concordant alignments, right? So you come to a locus, and unless there are X reads which have that exact same indel, it's not going to be a proposed indel, right? See what I'm saying? So unless you have 5 reads at the site, which all with the same indel, you're not even going to propose that as a possible indel, right? So that would be one reason why it wouldn't be called with the unified genotyper, but maybe it would be called with the haplotype caller, right? Because it'll show up in the assembly graph. You'll say it's a possible haplotype, evaluate its probability, and potentially it's an indel that you call. So maybe. Okay, so maybe let's go through a few of the slides. I'll show you how it works, and then we can keep talking. Okay, so basically what I said is that you're going to do this assembly graph thing. The paths in the graph are potential haplotypes that need to be evaluated. And then from then on, it's exactly the same as the UG, right? So you have these haplotypes. You evaluate them with the same HMM, the exact same code. And then you do all this stuff about determining the genotypes and computing allele frequencies. It's all the same as the unified genotyper. So this is what an assembly graph looks like. So you have these reads, and you're looking for overlap in the reads. And so here's a bunch of reads that overlap. And what you'll notice is that they're all the same until you get to the end. These reads have T, TA, and where these reads have CC, right? So there's a multinucleotide polymorphism in these reads. So TA goes to CC, and otherwise the reads match, right? So when you build this assembly graph, what you're doing is you're pulling out k-mers, and you're stringing them together. So in this example, we have three mers. And so you're just reading them off. GAC, you put it in the graph. And then you had the GAC goes to ACC. So you put that in the graph and make a link between them. And you just keep going. So you have all these things. And then when you get to this polymorphism, that's where the graph diverges, right? And if we had more coverage in this example, you would see that the paths actually come back together. And you form this little bubble, right? And the little bubble are the polymorphisms, right? So the way this would work is you build this assembly graph, and then you just read off these paths. In this example, it's very easy. You have 2 haplotypes. You have the haplotype with the TA part of the polymorphism, and you have the haplotype with the CC polymorphism. So you have 2 haplotypes. You run those 2 strings through the HMM, and you evaluate the probability of the data. Very simple. And like I said, you use the exact same HMM code that was used in the unified genotyper. What's beautiful here is that you have these transition probabilities, and those probabilities are exactly the base insertion and base deletion quality scores we spent so much time talking about yesterday, right? So you just plug them straight into the model. That's what makes it so beautiful. So now for the rest of the talk, I'm going to show you some examples of where the unified genotyper sort of makes the wrong decision, but the haplotype caller does the right thing. So what I have here is our most favorite sample, NA12878. I'm showing you some of her data. So in the top it's like high-seq data. It's very beautiful, clean, new, fancy data. But unfortunately, it's wrong. So you come to this location, and there's all these variation that's very near to each other, right? So it's very unlikely to be real because it's all clustered together. It seems unlikely. So but what happens with the unified genotyper is you come along, and because you're treating each locus independently, you come to this locus and you say, there's this beautiful C heterozygous SNP. Sounds great, I'll call it. Then you come to this one, and there's like an insertion, and it's also equally beautiful, so you call that one. Then you call this T, then you call this deletion, and then you probably don't call that C because there's only 2 reads. Probably unlikely to call it. But then you call this C, right? So you've now called 1, 2, 3, 4, 5 variants in this region, when in fact, what happened here was that it's a large, like a 20-some base pair deletion, and just the aligner didn't put that deletion in the reads, right? So when you come to it with the haplotype caller, you take all these reads in this region, you put them into that fancy assembly graph, and all that comes out is 2 haplotypes. It's very beautiful. You don't get all this junk. You get 2 paths in the graph. You get the reference path, and you get this path, which has this large deletion. You evaluate those probabilities, and you call the large deletion as a heterozygous event. And then we've done fancy validation to show that this is a real event, and so we're happy that the haplotype caller can now call it. What's up?
Does it generate the BAM files again, obtain it now?
Yeah, unfortunately it doesn't. Everyone asked me for this. I guess I'm going to have to work on this because everyone asked for it. The question is, so I'm showing you here a BAM file with the new indel. And so the question is, does the haplotype caller produce that BAM file or not? The answer is right now it doesn't. It produces a VCF file with this deletion in it. And you can, of course, use the indel realigner if you wanted and say, give me these reads with this proposed indel and have the indel realigner realign it. And it will probably do it, but maybe not because this is too big for the indel realigner. So actually, the indel realigner probably won't do this particular example. So probably what I'll do soon is create a mode where you realign the reads to the potential alleles and output the BAM, because people seem to like this. It's sort of a way to validate the results. So OK, eventually I'll get that done. But right now what you get is a VCF file with a 20-some base pair deletion at this locus. What's up?
So that's going to be very alignment specific, right? Like you were saying. But so you've done all this with BWA. The question, have you tried with other aligners to see whether you use Nova line or something?
Yeah.
They actually perform well?
So the thing is, so you're right. If there's some aligner that would put this 22 base pair deletion in the reads, then the unified genotyper would call it, because it's very easy. Because it would come to this locus, it would find that there's reads with these deletions, and it would be able to propose that allele and do the right thing, sure. But the problem is, like, there's no aligner out there that will always get it right. You know what I'm saying? Like, and it's actually very difficult to put, using Smith-Waterman or whatever algorithm these people use, to put a large deletion into the reads. It's quite difficult. So I don't know if that answers the question, but basically, you can't, like, you don't want to be in a world where you're always trusting the aligner to do the right thing, right? Any other questions?
It's just something that probably is going to happen. So insertions of the same size, of course, you're not going to get because the reads were actually not mapped.
OK. Yeah, so there is sort of a bias there. But I mean, for 22 bases, you know, it'll be in there, right? Because what will happen is, you know, the read will come so far, and then it'll start reading into the insertion. The aligner won't know what to do, so it'll just soft clip those reads, right? Or hard clip those reads, or whatever it does. Soft clips those reads, right? So what you'll have is you'll come to the read, and you'll notice that at this particular location, all the reads past it have soft clips. And you're like, oh, that's very strange. And that's actually one of the triggers for deciding if a region is variable or not, is an excess of soft clips, right? And so then you can simply unsoft clip those reads, throw them into the assembly graph, and find these large insertions. So you actually can call large insertions.
But for example, insertions which are larger than the
Exactly.
size of the read.
But as they get larger and larger, then you don't have any reads mapping in the area, and then you're right, you can't do anything with this approach. Exactly. So one benefit of doing a whole genome assembly and using like all the unmapped reads would be that you get these large insertions, right? Because you get the places where the reads don't have the correct approximal placement, right? Because right now we're assuming the approximate placement is correct. So places where it's not correct is where the whole genome assembly is beneficial.
Sort of along those lines, are you bringing in the if there are unmapped mates?
So we bring in unmapped mates. Basically, we bring in all mate pairs, so including the unmapped mates.
Okay, so you're using the pairing to maybe get even larger.
Yeah. I mean, so it helps some, but it's not going to get the huge ones, right? What's up?
Have you tried the unified genotype? Have you tried this map distributing?
Say that again. So if you use the unified genotyper to do what?
To align this reading to get a signal with the haplotype.
Well, so what you could do, so potentially if this deletion was in, like, for example, dbSNP or whatever your database was, you could use the indel realigner, and potentially it would realign those reads to that variant. So probably in this case it wouldn't, though, because the event's too large, and I think that there's some limit of like 10 base pairs or something in the indel realigner. So it doesn't allow like deletions of size greater than 10, I think, or something like that. Anyway, if this event isn't in your database, then you're sunk. There's basically nothing you could do, right? You could potentially it would know that clustered SNPs is a trigger, and it would do, like, you could, Eric mentioned that you could put it in this Smith-Waterman mode where it does this very extensive calculation, and potentially it could come up with this allele, but I think because of the size, 20-some base pairs, it's not going to come up with that.
Sorry, I just don't quite understand how you've gone from there to that. Are you saying that all those large blank parts are shifting over?
Yeah. So let's see here. So basically, what you see is that it's sort of a tandem repeating thing, right? So you got these alternate ACs, 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 and then switches to GTs, GTs, GTs. And so right here you have ACA, I don't know, whatever you have, TCT, I don't know. So somehow when you shift all of this stuff over to here, it matches up with the reference, right? I mean, we have to know, we don't even know what those bases are, so there's no way to figure it out, like, base by base, but because that's an insertion, right? So we don't even see those bases. That could be anything. But what it's saying is that when you put this part of the read over on this side, then it's matching the reference, and there's no mismatches, right? Just like with the indel realigner example you saw yesterday. It's just this is sort of a larger case where that happens, right? So these are the same reads. It's just when you shift that stuff over here, then it matches the reference. Make sense? Okay, I think I have a few more examples. So here's another example of a multi-nucleotide polymorphism that you can call for free now out of the haplotype caller. And so now I have a family, father, mother, and daughter. And apparently the father and mother have this multi-nucleotide polymorphism in the coding region. And the daughter inherited the reference alleles from both chromosomes. But what's interesting here is that what can come out of the haplotype caller is instead of two individual SNPs and two records in the VCF file, you get a single multinucleotide polymorphism. And this is incredibly important for people who are worried about the functional annotation implications of these changes, right? Because if you consider these changes independently, you're going to come up with a different answer than if you consider them in concert. And furthermore, what's very interesting is that this particular example, some pipelines, when they call it as individual SNPs, they actually mistakenly filter out the first SNP, but not the second SNP. Because you'll see the first SNP, you have something like 6 reds in a row here or something. And so many pipelines would filter that as a homopolymer, I don't know, there's some sort of homopolymer filter that they use. They would get rid of that SNP, and you would be left in your VCF file with just this one variant, which is totally different from what the reality is, right? So by considering these variants in concert, you sort of get a much more holistic picture of what's going on here. So probably everyone appreciates this because it's in the coding region. It's very, very nice. Any questions about that one? What's up?
Can you give a more precise definition for multinucleotide polymorphism?
What's that?
You give a more precise definition for multinucleotide.
Oh, so by multinucleotide, I just mean that it's not a SNP. It's a variant that is 2 changes, right? So what would come out of the haplotype caller is so the reference would be blue, red, red goes to red, red, blue,
so
right?
It has to be on the same haplotype.
Yeah, it has to be on the same haplotype.
What is the maximum number of variants within certain regions?
Let's see. So there isn't a fundamental limit. So what it does is it uses a likelihood ratio test for for whether the variants happen together on haplotypes or whether they're seen on different haplotypes. And if they're always seen together, then it would combine them together into a multinucleotide polymorphism. So it's based on, it's like an LD-based calculation that uses population information. Does that make sense?
So if two variants are on the same haplotype and are really far away from each other, I still can call them.
Yeah, you can. Yeah, you can.
Excuse me. How would you represent this in the VCF file? I don't know.
So by default, what would come out is a single record. That's an MNP record. So the ref allele would be blue, red, red, whatever those nucleotides are. And the alt allele would be red, red, blue. So you'd have 3 bases go to 3 bases. And of course, you can turn that off by default. If you want to have 2 individual records, you can. People get nervous when there's these strange records.
So this is going to be report each one of them in a separate line.
Say that again? It's going to.
Each one of the genotypes are going to report in a separate line. No, it's going to be reporting the same.
It's to be the same. It'd be 1 record. Yeah, it'd be 1 record. So your sample would be, this sample would be 0/1, where 0 means blue, red, red, and 1 means red, red, blue. What's up?
I was just following up.
Oh.
If you output or emit all positions for output, I didn't know. Like still on the VCF, how it's.
Oh, well then it would be just the 1 allele, the 1. So you would have, blue statements about just blue and then red, red. They would all be separate lines if you wanted to do that.
So it's going to be separate lines. There is no way to report in the same line to represent the haplotype itself, to show that they are linked together.
Well, so he's asking about a specific thing there. He's asking about emitting all sites, whereas if you run this thing in sort of a discovery mode, then you would discover this variant, and it would treat this as a single variant, and you would get one line. So that's the difference. Does that make sense?
So can you report the decision that would be referring to the platform?
The position would be the first position running left or right along the reference. Is this making people nervous that it's a MNP? I'm just surprised. Because of course, if you wanted to, you could output it as 2 SNPs. I just think you would get the wrong answer then. Or most people would do the wrong thing with a VCF file that had 2 SNPs. What's up?
What I've read online about haplotype color, some people have issues with the genome that they use. And
How do you mean?
like if they use their own particular genome, it doesn't work. But if they use the one suggested by GATK, then it does work. Are you aware of that at all?
No, I'm not quite sure what you mean. You mean like a different reference build or something?
Yes.
Hmm.
And I don't know what the differences are, but it's just.
Yeah, I don't know. I'm just not sure. I mean, let's see. What do I know? Who has run it in non-human? Let me think about that. Well, certainly we have users that are.
They were 2 different human genomes.
Just a different reference assembly?
Just a different reference.
Oh, I don't know then. I'm not sure. I mean, we use it in a variety of cases. We use it for all of our exome sequencing projects now are using it. We use it for 1,000 Genomes project. We use it. I mean, there are definitely non-human people that are using it. So I don't know. I mean, it is new and experimental, so I'm sure there's tons of bugs and things to worry about. But it's gotten a lot of use.
But those problems might not have been entirely specific to haplotype color. Some people sometimes have problems with they're using their own reference or different references because there's a number of steps that you need to do to have your reference work. Some people might think it's the haplotype color specifically, but it might be just it would have had problems with another tool too. So I'm not sure this was a specific problem.
For that example where the haplotype color found deletion, why wouldn't the local realignment
So I think the problem with local well, so first of all, local realignment would only find it if it was in the database of known polymorphisms, right? So if this variant's not in your database, it's not going to find it. But second of all, I think it's not going to find it because it's just too large. I don't think it's going to allow, because it uses this Smith-Waterman calculation, which has, you like discount for every deletion base, you're adding more discounted penalty, right? So you have a penalty for each deleted base. This is the gap continuation penalty. So you just sum up these penalties. And eventually your penalty is just so large that you say this is not it's not likely to be real. And so having a few mismatches is more likely than having a bunch of continuation penalties. It's just the way the mathematics works out, that it's just not going to allow larger deletions. So it's just the length of the event is the reason why. Okay, so I think I have one more example. So this one's crazy. Okay, so you come to this site. And what I'm going to tell you, you sort of have to just trust me, I guess. But what I'm going to tell you is that all these reads show the exact same thing, right? So what you see here is an insertion and C. And so what the insertion here is it's AC. So you have the bases TAC. And then here you have the bases TA followed by C insertion. So here you have TAC, here you have TAC. All these reads are showing the exact same thing. But due to the whims of the aligner, some of those reads got insertion C, and some of those reads got A insertion, even though they have exactly the same base content, exactly the same. The aligner just did something strange. It has just the whims of the aligner. And so what happens with the unified genotyper when you come to this site is that you propose this allele, this insertion allele, and you call it. You come to this site and you say, this is a crazy multi-allelic SNP, right? So you say reference goes to C or to A, and then you come to this site and you say that's a beautiful insertion. So you come to this site and you call 2 insertions and a multinucleotide SNP. I'm sorry, a multi-allelic SNP, right? You've called what is it? 4 alleles. You've called 4 alleles in this one site. When in reality, all this is a complex substitution. It's just TT goes to TAC. That's all it is. And so when you throw this into the assembly graph, you get out one path. TT goes to TAC, and you can call this as a complex substitution. So does everyone see how you can go from 4 alleles to one very simple allele? Okay. So the way the haplotype caller works is it's exactly the same as unified genotyper. You have inputs with your analysis-ready BAM file. You can control things about which quality score worth of variance to emit. There's a new parameter about the pruning in the assembly graph, which we sort of talked about a little bit. And then you output the VCF file. So the command line is very simple. GATK, you have your reference, you call the haplotype caller, you give it your BAM, and you have the output. And then you have this pruning parameter that you can use to control the runtime, right? So the way the pruning works is that's an old sentence. But anyway, it's basically it's the minimum k-mer length to include in your graph, right? So a pruning of 3 means that you have to have a k-mer weight of 3 or more to include it in your graph. So you had to have seen it with 3 or more reads essentially in your graph to keep it. So as you increase that value, you're pruning away some of the paths and you reduce the runtime. You lose some sensitivity by doing that, but you gain in lost runtime.
So what's the default value?
The default value for the pruning, it's actually very conservative. So the default value is 1, so as to not miss anything.
What is K?
Excuse me?
What is K, your k-mer size?
Oh, so interesting. So I didn't go to those details, but it's actually, so the k-mer size is every possible k-mer from, I think, a minimum of like 10 to the length of the read. So it's actually making many assembly graphs. It's using all possible k-mers. I really can't hear you at all, sorry. Oh, see, I knew you were going to jump straight to non-human things. I have no idea. You know, I've never even run it. I've never even seen non-human data, so I have no idea.
But right now we don't support anything other than deep work. instance, with haplotype color.
Oh yeah, yeah. So it makes a diploid assumption in the likelihood calculation. So yeah, I mean it's experimental. I'm sure it will work. And the principle is the same, right? It's just different kinds of data. You have more variation, so you have more paths in the graph, right? And it would just take longer, but you can evaluate the paths in the same way. And there's no fundamental reason why it wouldn't work. Any other questions?
When you have reads that are mapped to a couple of different locations, will it use those reads in the algorithm?
It's only going to use the reads that, if the primary mapping is within the variable region. So I guess it would use half of the reads, right? If it has this algorithm of like placing them randomly, then it would use half of the reads. Anything else?
What would we do for chimeric molecules?
It would not do the right thing. Right. That would show up as a path in the assembly. And so if there was enough read data to support that path, then it would call that as a variant. Otherwise it wouldn't. So if there are a lot of them, then it would have an issue for sure. I'm not sure what this is. Oh yeah, so we're trying to tell you how to evaluate the results. But really the answer is variant calling is complicated, and so how to evaluate the results is the rest of the morning session. So just pay attention, and that's how you'll know how to determine if the mutation calling works properly. And that's it. So we finished variant calling, and now we're going to move on to all the things that get done to make the variant calls analysis worthy. And that's it. Thank you.
Can the variable region include the paired read?
Yeah. So basically, you have this region, and it includes all the reads in the region and their mate pairs. So as long as one of the pairs is mapped in the region, then you're good to go. So, okay.
So it differentiates. So when BWA soft clips between, say you can turn on quality soft clipping versus if it indels towards the end of the read, it'll soft clip.
Yeah, yeah.
So it won't take longer when you have soft clipping by quality score.
So soft clip by quality score, are you talking about this Q2 specific thing or something different? So Q2. Okay, so the Q2 is like a special code by either BWA or Illumina or whoever. And so it doesn't use those, right? Because Q2 is like, what the probability is like less than 50%, right? So it doesn't use those at all. Yeah. But the other kind of soft clipping are like the evidence for a large event, and it would use those.
Is it possible to see all the graphs? I mean, instead of however you determine what's the best graphs, you can actually path by paths.
The paths in the graph? I see. So no, not right now. That's an interesting idea. So the question is about the assembly graph. It might be interesting to sort of just output the paths in the assembly graph, and you could rank order them by their likelihood. But that's not what it does right now. That's a good idea. That's interesting. That's a good idea. Anything else? Okay. Oh yeah, apparently I'm the next speaker too. So why don't we take 2 minutes, and then I'll get set up.